The overall goal of this screening is to find potential therapeutic drugs against blood cancers.
This method can help answer key questions in the drug development field.
The main advantage of this technique is that the system is composed of several well-established procedures.
Though this method can facilitate the search for therapeutic drugs against blood cancers, it can also be used to search for drugs to treat other cancers, such as melanoma and pancreatic cancer.
Begin this procedure by selecting a subset of heptamer sequences. The sequences can be random or designed to target specific cellular RNAs. To target specific cellular RNAs, use an appropriate computer program to find hairpin structures resembling the T-arm in a target RNA. Select complementary 7-nucleotide sequences immediately downstream of the hairpin. Using the phosphoramidite method on the controlled pore glass or CpG support, synthesize each of the selected heptamers as a fully 'O' methylated, 5' and 3' phosphorylated RNA with the DNA RNA synthesizer. Leave the 5' terminal DMT protecting group on in the last cycle of the synthesis. After completion of the last cycle, transfer the CpG with the synthesized oligonucleotide from the column to a glass vial with a Teflon-lined screw cap. Add 1 milliliter of 29% ammonium hydroxide and incubate the tube at 55 degrees Celsius for 8 hours. This will cleave the oligonucleotide from the CpG